Good day, this is the Professor, CBMD PhD, and today's Friday, August 18th, 2023. It is 4:11 and 10 seconds, a.m. Eastern Standard Time, and today's research, fusion in cancer. So let's hop to it here. Research raises hopes for new treatment of fusion-driven cancer. A new form of gene therapy can stop cell division in a particularly aggressive type of cancer. Date August 11, 2023. Source: Aarhus University. Using the so-called molecular scissors, CRISPR-Cas9, researchers have developed a gene therapy that can stop cell division in a subtype of the aggressive blood cancer, acute myeloid leukemia. Even though the study focuses on blood cancer, many other types of cancer are also driven by fusion genes. For example, some lung cancers and sarcomas. The researchers hypothesize that this technology can become a platform technology for specific types of cancer driven by fusion genes. And so far this gene therapy has been carried out in the laboratory on cell lines and mice, but the results are an important step towards developing a new form of treatment based on targeting the drivers of the disease. In brief, the researchers have succeeded in cutting genes that in fusion cancers are fused incorrectly together and which send a fatal signal to cells to begin dividing uncontrollably. When the fusion gene have been cut, the cancer cells stop dividing. In this study, the researchers have applied this treatment to cells in a laboratory setting by opening the cell membrane using electricity, a recognized laboratory method that allows substances to pass through the cell membrane. In this way, the scissors, the enzyme Cas9, can target the gene together with guides that consist of RNA, guide RNAs. You can use this method in the laboratory, but not in patients. We need to investigate how we can get both scissors and guide RNAs into the cancer cells. The scientific investigators and microbiologists are already working on using fat particles as a delivery method in a parallel study. This method worked very well as a delivery strategy for the mRNA vaccine against COVID-19. As so far the researchers have inserted cell lines with cancer in the flanks of mice. The non-treated cell lines grew into large tumors, while the treated cells that had been exposed to the gene therapy before being inserted into the mice resulted in much smaller tumors or none at all. The gene therapy seems to be extremely effective. In the laboratory, they don't know whether they can make the therapy effective enough to target all the cancer cells. There is a hypothesis within the field of cancer research that if you target the majority of the cancer cells with a treatment, the immune system will take care of the rest. The next task will be to demonstrate that the treatment with the most optimal delivery method also works in mice that have the specific fusion-driven subtype of leukemia. The researchers are aiming to examine whether this treatment strategy could be a new and better treatment option for other fusion-driven cancers. There is still some way to go. For example, the researchers do not yet know about the potential side effects of this gene therapy. One of the challenges is that if they cut the DNA in a cancer cell where the chromosome is placed incorrectly, then they'll also be cutting the same chromosome in other healthy cells. And since the fusion gene is not present in healthy cells, the theory is that healthy cells can repair their DNA themselves, but naturally they have to look into this more closely. This is still a stage of translational research, but it is a breakthrough because they succeeded in using CRISPR-Cas9 technology as target treatment for the specific fusion gene that drives the cancer. Even though there is still a lot of work to be done, the results give hope for an effective treatment for patients who have had limited opportunities until now. They hope that in the future they'll be able to introduce a more gentle and more effective treatment without serious side effects. As cancer occurs when cells begin to divide uncontrollably somewhere in the body, one of the reasons for this is the so-called fusion genes. In fusion cancer, genes fuse together incorrectly during cell division. Moreover, the fusion genes signal to the cells to divide uncontrolled and intensively, thereby driving the disease. In lung cancer, leukemia and sarcomas, and soft tissues are all examples of cancers with fusion-driven subtypes. There are also other types of cancer that are not caused by fusion genes, but rather other genetic abnormalities, including gene mutations. And this is the Professor, Professor of Microbiology, Bioinformatics, Pathophysiology, and Leader of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.